This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 782, recorded on July 21st, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York, Amy Rosenfeld.
Hello, Vincent. How are you?
I'm well. It's just stopped raining. It's nice out.
Good, but it was more exciting than the weather. Dixon came to visit.
Yeah, Dixon was just here in studio. In fact, Amy is next to me for those of you listening, because it's just us. And I said, what's the point of you being down the hall in the annex?
Well, the annex actually is being occupied by my lab.
The annex is Saul's office?
The annex was Saul's office, and then it moved down to the lab.
Okay.
And now and that's Edmund's Bay, and then on the other side is Michaela.
So normally you do, there's a Zoom station in there, but it's kind of noisy during the day with the lab and everything, right? So
We're busy. We're doing experiments.
Yeah, we're doing experiments. So I thought it would be best to bring you here. We have one mic between us, so hopefully I can make it sound good in post. But if it's a little echoey, that's why. And we thought we would proceed. Everyone else is at ASV, the American Society for Virology, which is the annual meeting. This year it's online. But I'm not there right now. I'm doing TWiV. I like TWiV. And thank you for joining me. Appreciate it.
My pleasure. You know, I was more excited to see Dixon than you, but you know.
Dixon he came up here in my office right here to get a photograph. The university wanted him and me photographed. He and I?
Yeah, it's he and I.
No, wanted him photographed and wanted me photographed. So it's him and me.
Okay.
For, I don't know, an article they're writing about TWiV or something. So Dixon volunteered to come in, and he looks pretty good, right?
He looks great. He's in great spirits.
Yeah, he's very pleased.
Very full of energy.
He said to me, you know, Vincent, you always say you have no friends, but you have one.
Me?
Well, he said to this as I was putting him in the car.
It's good.
Right. So anyway, it's great to see him. He'll be with us from now on. And Friday, of course, we have a ASV TWiV at 5 p.m. Eastern.
It's live streamed.
We are going to live stream it. We were thinking of live streaming this one, but there's a difference between live streaming and doing a podcast, right? On the live stream, you're kind of focused on the questions, right?
Yes, that's what we do it in the evening.
And then with the podcast, we focus on the papers. So for those of you who don't know, Amy and I do Q&A with A and V every Wednesday night, 8 p.m. Eastern, live streamed on YouTube. We've been doing it for 6 months.
Right? Really?
Not that long?
I don't know. It started like last November, and then we took a hiatus, and then we started back up.
No hiatus.
Yeah, we took like a hiatus. The hiatus.
Anyway, we answer your COVID questions or any virus questions, in fact. And we usually get between 400 and 500 people, which is awesome. And it's a little community. They chat among themselves. But it's hard to do a paper in that setting, right? Because a paper, you want to focus on discussing it and not have to go back and forth to questions. So that's why we're doing this as a regular podcast.
But you do a paper with Nels.
I find it difficult to do the paper. It's very distracting because there are a lot of good questions, and mainly Nels talks in TWiV, right? I don't do a lot of talking. And so I'm looking at the questions and trying to slot them in as appropriate. I find it harder. Not impossible, but harder. So, but anyway, tonight, Amy and I will have the live stream. So we get both worlds. All right, we have 3 papers, and we're going to get through them in maybe an hour, right? Because Amy's got plaque assays to do.
I do have plaque assays to do. I have 3 more trays, and I have to split several flasks to make trays for tomorrow.
Okay. You sound a little hoarse. What's going on there?
I'm going through puberty.
Oh. I thought that happened at like 12 or 13 or something.
I'm a late bloomer.
First paper, Nature Medicine, A Brief Communication. And by the way, this one, I think they're all picked by Amy. Well, she sends papers.
I drop them into
she drops them in the Dropbox, and then I look at them. And so this first one, immune responses against SARS-CoV-2 variants after heterologous and homologous ChAdOx / BNT162b2 vaccination. So that's adenovirus vectored spike versus and mRNA. People always ask, can I get mRNA after? This would be the AstraZeneca vaccine, right?
Yes.
And here are the data. Where does this come from? Let's see. A whole bunch of places in Germany.
Yeah, but it's really an English healthcare. It's really an English study.
It is. Look at, but all these universities, Hanover Medical School, Ulm University Medical Center, German Center for Infection Research.
Yeah, but it's really from the Hanover. Maybe I thought Hanover was in England, but it's actually in Germany.
Hanover, Germany. So the first author, let's see, there are multiple first authors. Yes, Joanna Barros Martins, Swanchi Hammerschmidt, and Ann Kosman all contributed. And Jorg Behrens is the last author. All right, so the issue here is that, as everyone knows, the AstraZeneca vaccine was released, and then what was it? March 2021.
It was paused.
It was paused because of clotting issues in some cases, right?
Yes.
And this is called vaccine-induced thrombotic thrombocytopenia.
Yes.
Or a simpler version, thrombosis with thrombocytopenia syndrome. Not much simpler, right?
Not really.
And they think it's induced by antibodies directed against platelet factor 4. So those antibodies are induced somehow by the vaccination? Is that the idea?
I guess so. But that's not really the point of the paper.
You know how I like to diverge, though.
I know, but we need to focus today.
Okay.
Today we need to streamline.
Put it down. Put it down. So the question is, if you got the first dose of the AstraZeneca, do you have to get a second AstraZeneca, or can you get an mRNA vaccine?
Well, it wasn't, can you get an mRNA vaccine or whatever it was. We're giving you an mRNA vaccine because we can't use this. What's going to happen?
And what's going to happen? Is it good or a bad thing to change vaccines?
And it turns out to be better.
Yeah. Are you surprised?
No, I'm not surprised. And I will tell you why I'm not surprised. If you had asked me a week ago, I would have said I was surprised. But I'm not surprised today because of an Ebola paper.
An Ebola virus paper, which said what?
No, an Ebola vaccine paper.
Yeah.
So that paper, they made 8 different viral vectored vaccines, all with the glycoprotein. They had some in Newcastle disease vectors. They had some in parainfluenza vectors. And they saw that all of those vectors, even though that they had the same antigen, resulted in some of them giving absolutely no protection to the non-human primates, some of them having IgG, some of them having really high GA, and various other things. So if you had asked me, as I said, a week ago before reading the Ebola vaccine paper, I would have said yes. But after reading that paper, no.
In this study, as you said, this COVID-19 contact study cohort of healthcare professionals. Actually, it's Hanover Medical School vaccines.
Yeah, I thought it was in England.
No, it's not England.
But
You're right. Okay.
It's Germany.
So the medical school vaccinees, the healthcare people who got a dose of AstraZeneca, they're offered a choice between AstraZeneca and BNT.
Yes.
So if you were offered that choice 6 months ago, what would you have done?
I still probably would have gotten the BNT.
Yeah, I would have too. I like the mRNA vaccines. And then they could study these two groups. So they had 129 AZ prime vaccinees without previous infection. And 32 of them chose to stick with AZ, and 55 chose to go to mRNA vaccine. And then they had, for comparison, this is pretty cool, 46 BNT vaccinated people. That's mRNA vaccine, right?
Yes, but you notice that the only one that they can't do is the where you got the BNT first and then the ChAdOx.
Nobody would do that, right?
Well, no, yeah, nobody has done that.
All right. So they looking at various antibodies in the sera of these people. So the mean anti-SARS-CoV-2 spike IgG and IgA had declined by 42 and 66% 30 days after the prime, which is maybe a little quick, right?
No, I think it's what's anticipated.
Similar to the declines in the mRNA vaccinated individuals. However, after the booster, they have both increased IgG and IgA in both groups, 11.5-fold increase for anti-S in the ChAd BNT, right? And 2.9-fold increase after homologous ChAd, so ChAd.
Right.
So going heterologous gives you a better boost.
Gives you a better boost.
Is that because the mRNA vaccine is inherently booster better?
No, because you just saw that they induced the same amount for the prime.
I suppose. But there's something about when you use it as a boost that really improves, right?
Yeah. As I said, with the Ebola paper, they used parainfluenza 5 and parainfluenza 3 and Newcastle, and they all had different, even though it was exactly the same antigen. So actually, I get that this paper is like a proof of principle, like you can get the heterologous, don't worry about it. But it's actually far more interesting to me in the fact that they never say this. It's the exact same antigen delivered in two different ways, and you get two different responses.
So the vector, the carrier matters.
The carrier matters.
Sure. And in one case, it's an adenovirus. In the other case,
it's just a lipid droplet.
Which is better, apparently.
After as a boost.
Yeah.
But how you're going to figure out that mechanism?
That's very hard, right? What would you do? I mean, you could use an animal if you replicate the finding, right?
Right.
So then what would you look for?
You'd have to, what would you put in? What would you do? Just have a lipid droplet of any RNA? Because it's the lipid droplet, right?
It's the adjuvant function of the lipid droplet, right? Yeah, I don't know. It's a good question. They also looked at the B cells having these, B cells have antibody on their surface, right?
Yeah, so they looked at different memory cells.
So they can pull them out of the patients with appropriate protein, right?
Yeah, they do capture. They do antigen capture. And they say that you have a different ratio of memory B cells. You have more memory B cells.
Yeah, so in samples from before booster, spike-specific memory were in 53% of the ChAd group, 43% of the ChAd BNT group. And these, they say, are a rare population of B cells. In contrast, spike-specific memory B cells were significantly increased in all vaccinees from both groups. In contrast to the antibody responses, the heterologous vaccinations led to expansion to a similar extent. So basically, the memory B cells are the same in both regimes, right?
Yeah, they eventually get to be the same. It's only the first.
And it's just the antibody level that's different.
It's just the subclass.
And then they did neutralization. So
this actually goes to something that Daniel is always harping on. There's recently very it's a point of contention for him is whether or not antibodies are correlative to immunity.
Your voice is fading.
Well, it comes and goes. That's what happens when you're going through puberty and the change.
He doesn't know if antibodies are the correlate of protection.
Yeah.
Well, I don't think anybody knows, right?
So that's something about this, right? Because you had all these antibodies, and then you find out that the memory B cell response is pretty the same, right?
I bet the protection is the same in both cases against disease. Right?
Against disease, yeah.
I mean, the titers don't matter,
Right. Well, people always think that the titers are important, right? We're always harping. Antibodies are waning. That's why we're going to, like, all these variants are going to kill us.
Neutralization using a surrogate, which means a pseudotype virus. Let's see. So 81 of 88 people had neutralizing antibodies against the original isolate, the Wuhan isolate.
Before the boost.
Before the boost. In contrast, antibodies against 3 variants, 117, P1, and 1351. Is that alpha, beta?
I think alpha, beta,
gamma were less frequent after the booster.
After the booster with the homologous vaccine, with Chad, but not after the booster with the mRNA vaccine.
So Chad and Chad mRNA equally boosted against Wuhan.
Yes, but the Chad did not protect against the variants.
Yeah, they had reduced neutralization, right?
So the breath, somehow the vaccine is resulting in a change of breadth of the antibodies that you develop. That's not clear to me how that works, that the vector would influence that.
Yep. I don't know either how it's presented. I have no idea what cells it ends up in. It's really a good question. But maybe it has a lot to do with the adjuvant. I don't know. The adjuvant of the mRNA vaccine.
I didn't think that there was an adjuvant in that.
Well, I think the lipid nanoparticle is an adjuvant, acts as an adjuvant. The lipid in the mRNA itself, probably, right?
Yeah, probably.
So you can get reasonable neutralization against alpha, beta, gamma variants. Of course, they didn't have Delta at this time to test, so they didn't test it. So booster immunization led to an increase of neutralizing antibodies in both groups, and the heterologous vaccination efficiently induced neutralizing antibodies against all tested variants of concern.
Oh, so the sky's not falling.
Well, it never was it?
No, Henrietta, the chicken little lady, thinks it's
Was her name Henrietta, really?
I think so.
And then he also looked at T cells, where they found no differences initially between after the first immunization, right? But after boosting, the spike-specific CD4 T cells increased in both groups, but were higher in the heterologous.
Yeah, and so were the CD8s.
And the CD8's the same thing, right?
Yeah. And you also saw more like interferon gamma and TNF alpha and stuff. Yeah.
And they noticed, so that's basically all the data, right?
Right.
They say here at the end, we started doing this before we had any information on safety and immunogenicity of this regimen.
Right.
And here, I think they also say there were no adverse effects of.
Right. There are no adverse effects.
And as you pointed out last week, we're already doing this with other vaccines in the U.S., right? Like Sputnik and mRNA vaccines.
Right. Anytime you come from another country that doesn't use an FDA-approved vaccine, they say to you, you're considered not vaccinated and you have to go get an FDA-approved vaccine. And most likely you're getting Pfizer or the Moderna. So not really clear. Like when everybody was saying, but it's not there's no study, there's no clinical trial. I was like, wait a minute, you guys, the left hand's not talking to the right hand.
Actually, they didn't collect data on safety and reactogenicity. They point out here, that's a limitation of the study.
Right.
But all they did was looked at immunogenicity. And the other thing they say, these are mostly healthy and relatively young healthcare professionals. So we can't generalize to elderly people or like me or specific patient groups.
Yeah.
And they couldn't test Delta.
Well, it came out before Delta. And they're not going to be able to test Epsilon and whatever the next one is.
Lambda's out already.
Yeah, whatever. I'm not good at the Greek alphabet.
Do you think we'll run out of Greek letters?
Apparently, that's what Dixon told me today.
So the real issue here is J&J mRNA/mRNA, right? Do you think it would be any different?
Well, apparently, according to today's data, it is. It's slightly less effective against the Delta.
So what is slightly less effective?
I don't know. Since there's no standardized test, I don't know what that means actually.
So you're talking about J&J/mRNA boost.
Yeah, well, just J&J alone is supposed to be.
Yeah, but that's a neutralization. As you and I know, it's not protection against disease.
Right. And I don't know, and since it's not standardized and one lab does it differently than another lab, I don't really know what
That's
it
what Ron Fouché said.
Yeah, but Theodore said the same thing.
Yeah.
Theodore and Paul said the same thing. And basically, Arturo Casadevall used that as an argument of why you couldn't standardize convalescent serum, or you couldn't test convalescent serum or something or other. And it was like, really? Did you think this thing through?
Well, you have to have a standardized assay first, right? Before you can test it. So he's right, because they didn't have a standardized assay.
Well, they still don't have a have a standardized assay. So it's a little, I don't know what a lot of this
And
means.
as Fouché said, who knows what level of neutralization correlates with protection, if any at all.
Right.
And I think you need to look at the T cell response as well.
Right.
Because isn't it the case that for some viruses, antibodies don't matter as much as T cells?
Right. So for this one, antibodies don't matter as much because you're not seeing patients with B-cell disorders coming down with more severe disease. Whereas like for polio, you see people who have B-cell or humoral response disorders. They're the ones who constantly are shedding virus, and they're more prone to like ECHO-11 and other entero infections.
Despite having intact T cell immunity, they still develop serious infections.
Yeah, some of them do.
So what you're saying is, for some viruses, T cells are important for recovery. For some, antibodies are important.
Yeah.
Shocking.
Yeah, I know. Luckily, I'm sitting down. A girl could hurt herself.
All right, next paper, Science Translational Medicine.
Yeah, this one just annoyed me.
Okay, let's see what annoyed you about it. Undiagnosed SARS-CoV-2 seropositivity during the first 6 months of the COVID-19 pandemic in the U.S. This is from all over the U.S., mostly NIH, it looks like, right?
Yeah, it's a lot of NIH.
Let's see if we had co-first authors. Here might have them, but I don't know. Heather Kalish, Carlene Klump-Thomas, and Sally Hunsbergers have daggers, so maybe they're co-first authors. And the last author is Caitlin Sattler. And so this is addressing the issue that
we missed a lot of people because we had people who were not very smart in charge of testing policies.
Yeah, we didn't test enough people.
No, it's that we only tested people who had symptoms.
Right.
Whereas, like, when Ori was in the lab with us at the very beginning of that pandemic, remember this story? I ran into Ori and his girlfriend, and she was the OB resident. And I said, why are you wasting all of the tests on the people who come to the hospital sick? You should just accept that they have COVID and move on. And you go out and take those tests and you find the people who are not presenting any disease and identify them. Remember that conversation?
I do. Well, vaguely. You have a better memory than I.
Well, because I had the conversation, but I'm sure I reported it back.
I'm sure. But they say here, look, it's difficult to estimate the true prevalence of disease during a pandemic. Extent of spread is only known after extensive study.
Yes, but if you're only testing people who come with symptoms.
But now we're testing more. Like here at Columbia, people are routinely tested, right?
Well, they have a random testing policy.
Yeah, it's still. So in the best scenario, what would you do? Would you test everyone once a week in the whole country?
At the beginning?
Yeah.
Yeah, for sure.
And now not so much because everyone is supposed to be vaccinated, right?
Yeah, not so much probably.
Okay.
But it depends on what population we're discussing. If we're discussing the vaccinated population, I wouldn't test them at all. If we're talking about the silly people who are not vaccinated, then I would still have the same testing policy.
But if you test the vaccinated people, then you could say, oh, the vaccine is only 60% effective at preventing infection. And you could get people scared.
Yeah, that's not so good.
Yeah, the point is that we don't care so much about infection. We care about protecting you against disease. Anyway, this is a survey of participants who had not been previously diagnosed. It was 462,000 people. And they did two different kinds of ELISA to look for antibodies against both spike and the receptor binding domain.
Yeah, but I would have liked them to have used spike antibody, done ELISA against spike and nucleic capsid.
I agree. And why do you think so?
Because that really demonstrates that you're infected.
Yeah.
The spike doesn't really demonstrate that you're infected. You could be just picking up vaccinated people.
So they didn't rule out vaccinated people in this study.
Well, they did because it was collected before the vaccines were released. But those people could have been randomly included in the trial, right? Because May 10th through July 1st, 31st, 2020, there was no vaccine.
Yeah.
But you could have been included in the trial and still participated in this. I just think it's scientifically better to have looked for two different independent proteins of the virus.
So they of these people, they enrolled 11,200 participants, and 9,000 samples were analyzed by ELISA. This is mainly U.S.
It's all U.S.
All U.S. population. They say it's representative. However, our sample was more highly educated and had higher employment rates, better access to healthcare compared to the general U.S. population. Okay. So this could be an underestimate.
Well, basically what they find is that we underestimated how many people were infected by a factor of 5-fold.
Yeah. So there were 304 seropositive participants, giving you 4.6% of the undiagnosed adults seropositive. And if you use this rate, we estimate there were 4.8 undiagnosed infections for every one that was detected. And they see regional variations, Northeast and Mid-Atlantic, the highest rates. The Midwest had the lowest seropositivity. This is early on, right? That's Urban areas had higher estimates, right? Which is expected. Right? Yeah, I mean, back then the suburban areas were saying, we don't even know what this is. We never hear of it.
Yeah, and the youngest group also.
The youngest, 18 to 44, had the highest seropositivity.
Because they have no symptoms. Again, if you just have a testing policy that you have to present symptoms, of course you're only going to get the people who have symptoms.
So why is it higher in females? 5.5% versus 3.5%.
Maybe it wasn't 50-50. Maybe they volunteer more. I don't know.
Maybe women volunteer more.
Maybe. I don't know. I'm not really.
But they could have been infected more frequently early on. Maybe they have front-facing jobs.
Yeah, I don't know. I'm not really that concerned about it.
African Americans was the highest, 14%.
Shocking.
Frontline people.
Well,
right?
They're most likely not the people who have the white-collar jobs.
Yeah.
Who were able to stay home. I mean, isn't that what Robert Furlove was talking about?
Robert Fully-Love. So in comparison to the national average, 4.6%, those who worked from home had a 3% seropositivity.
Yes, but the phrase before it says it has a lower seropositivity estimate. Once again,
when you're staying home, you're not going to be infected, right?
Exactly.
That makes perfect sense. Now, here it says here, those who reported prior vaccination for flu or pneumonia had a lower likelihood of undiagnosed seropositivity. What do you make of that?
So those who were vaccinated, they probably thought that those vaccines would give you cross protection because there was a lot of uproar about whether or not you should get a boost like with MMR and stimulate the innate immune response and protect you, right?
All right. But my take is different. My take is that these are people that seek out vaccines and they're careful about their behavior, most likely.
Yeah, but they had a lower diagnosis of.
Yes.
Yeah. I mean, I think
less infections.
I think that, it's fine.
You don't think there's any cross protection there,
do you?
No,
I just think that, yeah.
And those who had poor health conditions.
They're probably healthier people too.
People with poor health conditions associated with poor outcomes, heart disease, asthma, diabetes, they had lower rates of seropositivity. Probably also because they didn't mix as much with the population. Is that it?
I would think that they stayed home more.
Yeah.
more afraid. I know the people I know who are diabetic and had asthma were more afraid and stayed home.
So this equates to by July 2020, it's about a year ago, right?
Yeah.
There were 16.8 million undiagnosed infections in the U.S., in addition to the 3 million diagnosed cases. This is a funny, these suggest a higher level of infection-induced immunity in the U.S. population than previously predicted. That's, yes, if you got infected, right?
Yeah, but I'm still more concerned about the fact that we missed a minimum of 5-fold of the infections.
I bet it's even higher.
Well, yeah, that's what I said, minimum.
It's probably 5 to 10 fold.
Right.
But we did say that a year ago on
Right.
TWiV. We're probably missing at least 10 because Ralph Barrick said it initially.
Well, anytime that you're only testing people who have symptoms,
Yeah.
and you have your head in the sand that the disease is being transmitted by asymptomatic patients, and you continue to insist that you're only going to test symptomatic patients, you're always going to miss people.
Sure.
I mean, that's why that's not a smart policy. It's a bad public
They say here
policy.
the female higher seroprevalence may be that they get more asymptomatic disease.
That could be.
That could be. So if, let's say we had done this a year, we had done extensive testing of many people a year ago or more, how would that have impacted the outbreak?
Should have reduced the ability for it to spread as well.
Because we would put those people home and say, stay home.
They would be in quarantine. Yeah. And you would have flattened it even further, and maybe 600,000 people wouldn't. Well, apparently they're not dead. They're apparently all on the plane with Jeff Bezos.
You mean the rocket?
Yeah. Oh, it's a rocket. It's not a plane.
Apparently it's a rocket, yeah.
Whatever. Whatever. Apparently, according to
So why
people
didn't we test more people back then?
I don't know. I'm not in charge.
I think you have some ideas, no?
I do have some ideas, but
I think many people who were in charge of public health responses didn't realize the extent of asymptomatic infections. Is that correct?
I think that they were fully well aware of it.
But so
I think that manufacturing of a test was very difficult.
Yeah.
And you were led by people who were incompetent, who kept insisting you can't test unless you came from, you had some kind of contact with somebody from China.
Right.
Or you had symptoms, right? And then those people, then it became so polarized that you still couldn't test. And it was, yeah, I think it's a reflection of the person who is in charge of the country. Don't test because you're going to find symptoms. You're going to find more infected people. That's why you have so many people who died unnecessarily.
Well, also remember that even if the CDC had wanted to test more, they can't make states test more. They have no jurisdiction.
No, I understand that.
Right.
But they also did not have a great test. Took them like months to flush out their test.
Right.
It's not that hard.
No, it should never have happened. They should have let you do it, actually. You could have.
I don't want to do it. But
I know you don't, but you could have done it very quickly.
Right. But I also would have tested everybody. It should never have been a polarized thing. I mean, I had a different perspective.
I agree. I would have tested more people at least once a week. Hey, maybe we could have had a $1 a shot test.
$1 was too expensive when I did the math in October. It's too expensive today. But yeah, I think that there should have been a repercussion to the person who said, I have a lick-a-stick for $1 a day, for $1 a test, and never had a lick-a-stick. And he knew he never had a lick-a-stick. It was really kind of upsetting. And the fact is you could still use it today in other parts of the world, or even here in this country in pockets that are being poorly vaccinated, like Mississippi and Arkansas and Missouri and other places. And what are you going to do when you have to go back, send your kids back to school?
That's the key. School would really benefit from a cheap, frequent testing, wouldn't it?
Right.
Yeah, especially since we can't vaccinate.
Well, not yet.
Young kids, yeah.
Yeah, and they're not going, even if you were to say that they are in the process of getting all the paperwork together to ask for an expansion of the EUA, it's not going to occur until many places have gone back to school. So, for instance, my godson goes back on my birthday, which is in August.
Early August.
Yeah. So his sister will not, while he's vaccinated, his sister will be going back unvaccinated.
Now,
and will be there for many months.
And won't be tested likely.
No, they live in Florida. They're not going to be tested.
So Daniel would argue that we do have rapid tests, but they're not cheap enough.
My cousin cannot afford a $30 test a person.
Yes.
It's not happening.
Agreed.
And there's no reason for her to spend that amount of money.
Yeah.
It's a shame. Hopefully my next pandemic won't be as bad as this one.
You think people are going to remember this by then?
Oh.
No, they're not. They're going to forget. Public health has a short memory because it's all wrapped up with economics and.
I know. I listened to Larry Carr. I know. And what do I say about this pandemic? It's the worst one I've ever lived through since the 1400s of my bubonic plague years.
What about the 2009 influenza pandemic? You lived through that, though.
I did, but it wasn't really that.
No, it wasn't that bad.
Really a pandemic to the.
Technically it was a pandemic, but the virus was not as lethal.
Didn't
there were fewer
transmit
deaths
as efficiently.
All right, we have one more paper. This is from Nature. Broad sarbecovirus neutralization by a human monoclonal antibody. Oh, a lot of authors on this one from all over the place. You know, we got University of Washington, Pasteur Institute, Fred Hutch, many others. And we have some names of people we know. Let's see, the first author. Okay, we have equal contributions. Alejandra Tortorici, Nadine Nuchowski, Tyler Starr, and Roberta Marzi. And this is from, I told you where it's from. Some people we know on this paper, aren't there, Amy?
Yeah.
We have Amalio Talenti, I know. Skip Virgin. Wow, look.
Well, it's his biotech company.
Which one?
Vero Bio, whatever it is.
Ah, okay. Then we have Sean Whalen.
Mm-hmm.
We have Jesse Bloom, who I'm guessing did the spike mutagenesis.
Yes, yeast.
Those are the people I know. Someone's calling me from Columbia, but I'm not answering.
Well, David Vessler is at Washington and with Jesse, and he's done a lot of stuff recently too. That structure.
I'm sure many of these people are well known, but those are the names that jump out at me. All right. So this is up your alley because it's all about antigenic sites, and that's what you're working on these days, isn't it?
It is. I like antigenic sites.
It's funny because...
and cross-reactivity is a big topic now, too.
Yeah.
They talk about it being able to be cross-reactive against a lot of Serbecos, but not like the MERS coronas. Very interesting cross-reactive.
So once a week, we take antigens to the mouse house and we inoculate mice, and that's part of your inquiry into cross-reactivity and what is it, enteroviruses, right?
Yeah. Well, it's not just antigens. I take a live virus and I stick it into
infectious
the mouse.
Virus, right?
Yeah.
Is it infectious by the time we stick it? No, I don't think so. I think you inactivate it, right?
No, I don't.
Well, that's why it's an ABSL2, right?
Right, but if we all know that they're different by treating polio with formalin, the antigenic sites are different than they are from Sabin.
Right.
And from Mahoney.
Right.
So, no, I didn't want to inactivate.
So here they say, by the way, the receptor binding domain
is spike 2.
Is spike 2. The antigenic site 2 generates most of the neutralizing antibodies, but apparently, according to Theodora, not all of them.
Well, it was never thought to be all
that
of
one
them,
paper we did last week said it, but that's not correct. Apparently.
No, it's not. There's a huge amount of neutralization against the N-terminal domain of spike, which nobody, I think, really fully understands how it blocks. So maybe it blocks receptor interaction also by being sterically hindered. I don't know.
So there's a site on the RBD, site 2, that has been shown by others to give rise to cross-neutralizing antibodies. But this is apparently hidden.
Yes, it's cryptic.
It's cryptic. So in this paper, they describe a monoclonal against a site in site 2 of RBD.
Yeah, that you only see when at least 2 of the 3 of the trimers are open.
Which means they have to have bound to ACE2 already,
Yeah.
right?
So
Okay.
2 of them have to be open in order to see this site.
So this is from a B cell of a patient,
Yeah, but we're not doing it this way.
but you're doing this in general another way. But if I said to you, Amy, go get me B cells specific for SARS-CoV-2 from this patient, how would you do it in general? Not by your technique, but by what they did.
So they pulled out they cloned out the B cells. So they had multi-well plates and they put single cells in each well.
Right.
And then they took the supernames and asked which ones had neutralization ability.
They didn't pull out the B cells with an antigen?
No.
No.
They did not do antigen capture.
Okay.
Not the way we're doing it.
So they pull out a monoclonal from a B cell of a convalescent patient, which they say cross-reacted with 25 out of 26 glycoproteins representative of all sarbecovirus clades, the beta
Right.
coronaviruses, of which SARS-CoV-2 is a member.
And
right?
SARS.
And SARS-1.
Right, but not MERS.
Not MERS. Some of the human common colds? No.
No, because it doesn't do it. Well, yeah, I think some of them are. But not OC43 or...
Yeah, HKU1 and OC43 did not bind. All right, so then they looked at this cross-reactivity. They did ELISAs with
I would
RBDs.
have liked them to do it by surface plasma resonance. You get better numbers. I'm into that now.
That's your latest thing, surface plasma.
No, I'm back into this.
So tell people what is SPR.
A Biacore thing is?
You say, what's a Biacore? Yeah.
It's the machine that detects the kinetics. You put something bound on the gold chip, like your antibody, and then you, at a very specific flow rate, you flow a solution that contains the antigen over it, and then when it binds, it has like a weight. You measure the weight.
There's some change in refraction of
right.
Of
not
light.
weight, but yeah, refraction.
And so
of
you
light.
can measure the binding based on the refraction.
Yeah, and then you come up with KDs and K-ons and K-offs and everything.
We did this years ago for polio and its receptor, right?
Yeah, with Brian.
Brian McDermott.
But now I'm back into it.
That's good. So they say this monoclonal bound all the RBDs except the 2 BAT RBDs, ZC45 and onlong 112.
Yeah, but what's interesting is the broad recognition of S from non-Asian bats. Clades.
Yeah, clades 2 and 3, yeah.
Comes, and then Jesse has a paper yesterday on bioarchives about how they look at the evolution of the ACE2 binding of SARS-CoV-2 or of spikes.
Yes.
And they say, oh my God, non-Asian coronaviruses or coronaviruses from non-Asian bats also are able to bind ACE. Really? Shocking.
This is what you've said before, right?
Yeah, I think, and yet a certain individual is still pushing the idea that the virus came from a lab.
Look, they do SPR.
Yeah.
Surface plasmon resonance, nanomolar to picomolar affinities. And it's very good binding, right?
It's really good.
And they say this is an unprecedented breadth of binding of the antibody. And this binding is unaffected by changes in circulating isolates.
Yeah, because it doesn't affect the cryptic site.
They have all these amino acid changes at B117, B1351, B1427, 429, P1. No change because those changes are all at the dominant site.
Right. They're not in the cryptic site.
Yeah. Neutralization potency using authentic virus as well as pseudotype virus. And it's very good neutralizer with wonderful IC50s, the 50% inhibitory dose.
I wish some of our antibodies would be that.
And they made pseudotypes with the variant spikes, and it neutralizes all of those really well. It neutralizes a broad panel of ACE2 binding sarbecoviruses, including SARS, pangolin guangdong, SARS-2,
Yeah.
RATG13.
Yeah.
SARS-CoV, Wuhan Institute of Virology 1 and 16, and RSSHCO14. Those are all bat viruses, right?
Yes, but RS means it came from a different bat species.
Yeah, from RA, right?
Right. It's not whatever the A is.
Rhinolophus phoenicus as opposed to Rhinolophus something else.
Yeah, exactly.
So what's the structural basis for this? They do cryo-EM of
They
the
do
spike
both.
and
They
an
do
X-ray.
they do crystallography too. So in parallel, we crystallized it.
They did both. That's good.
Yeah.
That's what you can get when you have 50 authors on a paper. You can do cryo-EM and X-ray
I don't want
crystallography.
50 authors on my paper, but I mean, I don't mind them, but this is my plan.
So
right?
the picture shows that the antibody binds a cryptic epitope with an antigenic site 2 that was previously known. And its binding requires opening of 2 RBDs to grant access to the
Two of
FAB.
the three, just what I said.
So let me get this straight. So let's say the spike is bound to ACE2. Now you have 2 of these sites exposed. The antibody can then get in there and still block infection?
Yeah.
So it's not really blocking attachment, is it?
No, it's blocking fusion. I don't believe that they call it an attachment blocker.
So this epitope is conserved in multiple isolates, and the RBD changes don't affect it. They're clearly different in the structure, right?
They actually say the antibody binding to RBD does not affect or prevent recognition.
By other antibodies, so those would block attachment.
Right.
And so this is not interfering with so by extension, it's not interfering with attachment, right?
With attachment.
Competition binding assays suggest that serum antibodies directed to the site 2 are subdominant, which would explain why we don't have a lot of cross-reacting antibodies, right?
Mm-hmm.
And they could be used in combination with other monoclonals, right?
Yeah.
So this one, which seems to be very potent. But the real cool part, I think, of this is they look at escape. In other words, could we make escape mutants to this monoclonal?
Right.
It's a good question, right?
Yeah.
So they use deep mutational scanning with a yeast display RBD library, which Bloom published before. That's what we did on TWiV, I think, right?
Yeah.
So they basically mutate the RBD and they produce the protein in yeast, and then they can do things bind to it,
right?
Including ACE2 or monoclonals in this case, right?
Right.
So they could say, we could change every amino acid.
But one.
And see which ones escape binding.
Right.
Because you're not depending on making infectious virus. You're just making the protein here, right? And they say, only a few amino acid changes reduced binding to the monoclonal, and site 504 was the biggest effect, right?
Right.
Had the biggest effect in reducing binding. And 504 doesn't show up in any of the variants of concern, right? Which is amazing.
Well, because it's hidden most of the time.
Yeah.
Those variants of concern are really more exposed, solvent exposed.
So substitution of this position, G504, glycine at 504, with most other amino acids reduced binding compared to wild type.
Mm-hmm.
And so then they decided to try and so they passaged a pseudotype to try and select viruses that are resistant, right?
Mm-hmm.
So they passed VSV, SARS-CoV-2 in the presence of the monoclonal. You get variants emerging. And where do you think the change is in the variant?
504.
504, G504D. That's the only one in 18 individual plaques.
Yeah.
I think you can identify with this experiment, can't you?
Yes, I can. I can.
And the fitness is not impaired.
No.
So that means if this arose in the population, and it is bound to arise if we use this monoclonal, it will be fit, as fit perhaps as the wild type virus. It's not going to be fizzling out. And they have another antibody that targets site 2A, which is the dominant site, right? they easily got 6 escape mutants at
Mm-hmm.
5 different positions. So then they look in all of the genome sequences available as of May, SARS-CoV-2, 0.002%, 27 out of 1.3 million sequences have this change.
Yeah.
So I would say that's bad news, because if you start treating people with this antibody, those viruses are going to be selected for, right?
Sure. I mean, I don't know that I would say it's bad news, because the epitope is very hard to find.
It's bad news because the resistant mutant already exists.
Well, all of the resistant mutants already exist.
That's true. You're never going to find a monoclonal to which you're not going to get resistance, right?
So then what's the question?
Well, here you could use this in combination with Site2A antibodies, right?
Right.
I think that would be the way to go. You agree?
Yeah, I agree. I'm not, I think that probability is pretty low. And then they show it protects in their hamster model of disease, of infection.
So yes, they have a hamster model where they infect hamsters. What is it? Intranasally, right?
Mm-hmm. And look at what they find might be responsible for the lack of Fc gamma.
Wait a minute, that's first here, right? Isn't that? What is this talking about here? Oh, let's go through that first. So,
is this another topic I'm interested?
So S2X259 binding efficiently promoted shedding of the S1 subunit. So the epitope is on S1?
It's in the receptor binding domain,
which is S1.
Right.
So when monoclonal binds, S1 falls off, assuming that it's cleaved, right?
Right.
And they say this shedding might be responsible for the lack of Fc receptor activation. So some monoclonals could bind the virus and bind an Fc receptor and get pulled into cells.
Right.
Is that the idea? But this is not going to do that because S1 falls off, right?
Right. So you're not going to see ADE.
And they actually tested that, right? They did some assays, antibody-dependent phagocytosis and cytotoxicity, and they say,
we don't see any.
We don't see it. But if we make an uncleavable spike, then we see it, right? It's kind of a cute experiment, right?
It's a very good experiment. Cute. But this is another thing that I'm really interested in, right? Mm-hmm. It's the role of the Fc gamma receptors and whether or not your antibody works or not.
So they say that these data show that the primary mechanism of this antibody is blocking attachment to receptors through competitive inhibition of ACE2 binding. So it must be blocking after the 2 RBDs are already attached in some way.
Right.
Maybe it dissociates them because it has higher affinity than ACE2 does, right?
Maybe. Well, you haven't competed it off with soluble ACE2.
You're saying that's something they should do?
Yeah.
Yeah. That would address that, right?
Yeah.
All right. So then the experiment of the hamster. Yeah, they give virus. They give the antibody intraperitoneally, and then they do an intranasal challenge. Mm-hmm. So the IP is so that it's going to slowly diffuse out into the periphery, right?
Right.
And then they collect the lungs and look at virus. And so the administration reduced the amount of RNA and infectious virus by 2 and 2.5 orders of magnitude compared to the control monoclonal. Complete abrogation of replication observed in the lungs, given 5 and 25 milligrams per kilogram. Therapeutic administration, which is post-exposure, reduced RNA by 3 logs and blocked replication in the lungs of all animals but 1.
Yep.
Then they did the same experiment with B1351, which is the beta.
Yeah, could be.
South African.
It's too hard for me.
I'm still better with South Africa.
You can't, but that's not a good plan. But the numbers were fine. I don't mind them numbers, but You got to stick with one.
Reduce the antibody, reduce the amount of viral RNA, and abrogated replication in most of the animals. This is the variant, the beta variant.
Right. And the animals that got cocktails between this and another monoclonal outside of the site did even better.
S309 outside of this site, right?
Yeah.
And none of them got sick.
Nope.
We have no pathology. None of them got sick. So this is a broadly neutralizing and very effective monoclonal. It's also only one amino acid change apparently can give rise to resistance.
But it's hard to generate that change because the site is not solvently exposed.
Yeah. So they're saying maybe we could use this in the future for another spillover, which is going to happen, right?
Yeah. We could use it for a short time in the future until we get better drugs.
Yeah, I mean, this is not a solution to a pandemic, is it?
No. But we don't do solutions.
Let's think of this. Let's say someone gets sick and then they can't breathe, so they go to the hospital. It's too late to give them this monoclonal, right?
Yeah.
And if they're mildly sick, they're never going to get checked, and they'll never get this monoclonal.
Well, they're using monoclonals now, so somebody is going to the hospital or going to get checked when they're mildly sick.
Isn't it at-risk people, if they have a positive test, they immediately get monoclonal?
Yeah.
But again, in an infusion center, they don't have to go to a hospital, right?
Right. But again, it's a very select population.
Yes.
Which is not really the great way to do it, right?
Yeah, I agree. This is not a solution for the entire pandemic. This is to treat a select population.
Yeah,
right?
but that's like 3%.
But
that's like arguing about 3% of the world having all the wealth.
I don't like that. Do you?
No. So, but I would never, so this would not be my healthcare policy.
I mean, I think from a scientific viewpoint, it's a really interesting monoclonal, right? The biology is very interesting. But for treatment of disease, I think it's entirely impractical. And to get excited about it, I don't think is useful because it has to be infused probably intravenously. Maybe if they're lucky, they can do intramuscular. It takes a couple of hours.
Well, what is the thing that Daniel has?
And then it only lasts a couple of months at the best, right?
Yeah, but I think like when you're in the beginning of a pandemic and you have nothing else, this is fine.
But don't we have to test people more and see who's infected and treat them?
Yeah, but we don't do those kind of things.
Do you think this should be brought through clinical development and stockpiled?
Yeah, it's potentially possible.
Because they say this is a candidate for clinical development, could be stockpiled as part of a pandemic preparedness toolbox.
Yeah, it's part of a toolbox. But the truth of the matter is it's for like the first 3 months when you need to figure out what you've been exposed to and how you're going to deal with it. And then you still need to have better diagnostics and then development of pathogen-specific treatment, right?
I still think a vaccine is the way to go. So the question is, how could you induce antibodies in a better way to this cryptic epitope by vaccination?
Well, if they're all prefusion, right, they're not going to be open.
So they suggest just the RBD alone. We propose RBD-based vaccines could better promote, the elicitation of these antibodies.
But that failed. Wasn't Peter Hotez's vaccine an RBD only?
Yeah, but I don't know what happened to his vaccine, frankly.
And was Novavax a pre-RBD only?
Novavax is Spike, isn't it?
I don't know. I'm asking. I don't keep track of these things. I have other stuff to keep track of.
Novavax. We did that. We had.
What's his name? Matt Friedman on.
By the way, it was 90% efficacy in phase 3 and 100% protection against moderate and severe disease.
Great.
That was just last month. Let's see if this. I think it's Spike, frankly, not just RBD. What is Novavax?
Well, I thought that there were RBD only vaccines that failed.
Well, that was a Pfizer mRNA was RBD only.
Oh, the Pfizer mRNA, then I take it back. It's doing perfectly fine.
No, sorry, the Pfizer RBD mRNA didn't get pushed forward. The full spike did.
Oh.
But
as I said, I thought, then I'm getting very confused. As I said initially, I thought that the RBD only vaccines were not the way to go.
I agree, but these guys are saying there must be a way to use them to get these kinds of broadly neutralizing antibodies
but
induced.
The thing is that I think that it's going to be very hard to make this.
Yeah. It's full spike. Full spike, yep.
I think it's going to be very hard to make an RBD only vaccine. Number one, just because, as I said, it's in my mind that's already failed. And all of the vaccines that are very efficient are efficient because they're in the pre-spike, the pre-fusion conformation, which would make this epitope occluded.
Well, maybe you can make smaller versions of the RBD that make this exposed. This epitope.
I don't know, if you make too small, then you've ruined the structure.
Yeah. Well, I think it's something you could explore, right? It's worth exploring, maybe.
Yeah, sure.
Because I think there will be a lot of development in this area going forward. Anything else you'd like to say about this before we leave it?
No, I'm good.
You like this paper? Do you like it better than some of the others we've done?
Yeah, I like it better than some of the others. I think it's more comprehensive. And it's just happens to fall in quite clearly with the goal, with the framework of our daily research, which was good, right?
Yeah.
Plus, EVs are the next, our EVs are the SARS-CoV-2 of the EV world.
So our lab works on enteroviruses, right?
Right. And the ones that we're particularly interested in are the SARS-CoV-2 of the enterovirus world.
Which ones are those?
The ones that we work on.
All right.
The ones that we're interested in.
All right.
68 and 94 and 111 and various other things. They're the ones that we're interested in, right? And that I've said are now the SARS-CoV-2 of the EV world.
Stay tuned to Amy's work.
Well, I don't know. It's going to take a while. We're slow.
Yeah, 14 months.
14 months?
That's what you have.
Oh, not so good. Not so good. I need more people.
All right, that's TWiV 782, a special, efficient, focused midweek TWiV.
Yeah, we like focused efficiency.
The show notes are at microbe.tv/TWiV. If you have questions or comments, TWiV at microbe.tv. And if you like what we do, consider supporting us. We have this new venture called The Incubator, an office/studio in Midtown Manhattan, where we're going to be producing shows, and we'd love your support to move that forward. Microbe.tv/contribute. Amy Rosenfeld is here at Columbia University. You can find her work at enterovirus.net. Thank you, Amy.
My pleasure. We keep saying that, but I never update it.
Well, it's there, right?
Yeah, we need to update it.
And people can see your sparkly shoes there, right?
Yes, they can see my lovely Manolos.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.